Hi, this is the second biochemistry lecture. We are gonna to continue what we have already started, which is glycolysis. We are going to have an overview for the enzymes and substrates involved in glycolysis. Then we are going to go through each reaction in detail. First, we need to phosphorylate the glucose to glucose 6-phosphate by hexokinase or glucokinase enzyme. We require this phosphorylation in order to trap the glucose inside the cells to be able to continue the glycolysis. This 6 stands for the carbon atom to which the phosphate group is attached. As you know, glucose is made up of 6 carbon atoms. So, phosphate is attached to carbon number 6. Then glucose 6-phosphate is isomerized to fructose 6-phosphate by phosphoglucose isomerase. Fructose 6-phosphate would be further phosphorylated by the phosphofructose kinase 1 into fructose 1,6-bisphosphate. Then fructose 1,6-bisphosphate is cleaved into 2 molecules. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both of them are made of 3 carbon atoms. Actually, fructose is made of 6 carbon atoms. This reaction is catalyzed by aldolase. The glycolysis cannot continue unless it has 2 molecules which are the same. But we have here 2 molecules which are not the same. We can overcome this problem by isomerization of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. This reaction is catalyzed by triose phosphate isomerase. And now we have 2 molecules of the glyceraldehyde 3-phosphate. I got the first one from the cleavage of the fructose 1,6-bisphosphate and the second one from isomerization of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. And so all the following steps will be duplicated. Now, glyceraldehyde 3-phosphate will be converted to 1,3-bisphosphoglycerate by a glyceraldehyde 3-phosphate dehydrogenase. Then, 1,3-bisphosphoglycerate will be turned into 3-phosphate glycerate via phosphoglycerate kinase. Phosphoglycerate mutase is going to convert the 3-phosphoglycerate to 2-phosphoglycerate, and then from 2-phosphoglycerate to PEP via enolase, and finally we get our end product, which is pyruvate from PEP by pyruvate kinase. Now we are going to go through each reaction in details. First, phosphorylation of glucose to glucose 6-phosphate. This means that I need a phosphate group to be added to glucose. I got this phosphate group from ATP, leading to production of the first ADP molecule and to the loss of the first ATP molecule. Then, glucose 6-phosphate is isomerized to fructose 6-phosphate, which will be further phosphorylated to fructose 1,6-bisphosphate, and again I need a phosphate group, leading to production of the second ADP product and the second loss of ATP. Then fructose 1,6-bisphosphate will be cleaved into 2 molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which will be isomerized into glyceraldehyde 3-phosphate in order to be able to continue the glycolysis. Again, we have 2 molecules of glyceraldehyde 3-phosphate. Please remember this point. Now, the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate is done via 2 steps. Oxidation of aldehyde group of glyceraldehyde 3-phosphate. Oxidation means removal of hydrogen, which is equal to dehydrogenation. This means that glyceraldehyde 3-phosphate loses hydrogen. So I need a hydrogen acceptor to accept this hydrogen. My hydrogen acceptor here is NAD. Accepting hydrogen means reduction. So NAD would be reduced to NADH. And this is the first oxidation reduction reaction of glycolysis. Then I will add phosphate group to carboxyl group of the glyceraldehyde 3-phosphate. This phosphate group is linked by high energy bond, so that my new product, which is 1,3-bisphosphoglycerate, contains 2 phosphate groups, one of which is linked by high energy bond, so that this product is going to lose that phosphate group to give me 3-phosphoglycerate. This high energy bond would be used in this step to synthesize ATP, leading to production of ATP from ADP. This is known as substrate phosphorylation. Why? Because the synthesis of ATP from ADP is caused by the addition of phosphate group, which is derived from my substrate. So ATP synthesis is not done by the help of ETC. Please remember here, we have gained 2 ATP molecules because everything here is duplicated. Now, 3-phosphoglycerate will be converted to 2-phosphoglycerate. How? As the phosphate group will be shifted from carbon number 3 to carbon number 4 of the phosphoglycerate. 2-phosphoglycerate will be dehydrated, which means it loses water. The dehydration leads to something known as redistribution of energy, causing my new product, which is phosphoenol pyruvate, to contain high energy enol pyruvate phosphate. That's why it is known as phosphoenol pyruvate. So that during the conversion of PEP to pyruvate, this high energy enol phosphate would be used to synthesize ATP, leading to production of again ATP from ADP via substrate phosphorylation, and here there are 2 ATP are produced. As you know, glycolysis is made up of 10 steps. 3 of them are irreversible, while the rest are reversible. In this concept map, the irreversible reactions are highlighted by red color, while those reversible are highlighted by blue color. Let's have a look. Phosphorylation of glucose to glucose 6-phosphate is irreversible. Phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate is irreversible. And finally, PEP conversion to pyruvate is irreversible. Now, I need to regulate these 3 irreversible reactions. Why? Because if any mistake is done, I will not be able to go into the opposite direction. So my body needs to regulate the three irreversible reactions very well. Regulation. Those highlighted with blue are regulated, and those highlighted with red are not regulated. Phosphorylation of glucose to glucose 6-phosphate is irreversible and so regulated. Hexokinase is inhibited by glucose 6-phosphate. Why? Presence of high concentrations of glucose 6-phosphate means that a lot of glucose was phosphorylated. So, more ATP would be produced. So I no longer need glycolysis, I have to stop it. And here I stop glycolysis by stopping phosphorylation by inhibiting hexokinase. Then, fructose 6-phosphate. Phosphorylation to fructose 1,6-bisphosphate is allosterically regulated via inhibition or activation of phosphofructose kinase 1. The enzyme is inhibited by ATP and citrate. Actually, high concentrations of ATP mean that the cell contains enough energy. I no longer need glycolysis. Also citrate. Citrate is an intermediate with TCA cycle. Presence of high concentrations of citrate mean that high concentrations of ATP would be produced and I no longer need this glycolysis. However, the enzyme is activated by AMP and fructose 2,6-bisphosphate. AMP means the cell is derived of energy. I need to synthesize ATP. So I will activate glycolysis to get ATP, which would be used in energy requiring processes. I know 2-phosphoglycerate conversion to PEP is reversible. However, it is regulated since inulase is inhibited by fluoride. Finally, PEP conversion to pyruvate is inhibited by cyclic AMP. How? During fasting, my blood glucose level decreases, leading to increased secretion of glucagon by pancreas. Glucagon is going to bind to specific receptors, increasing the synthesis of cyclic AMP. Cyclic AMP would lead to phosphorylation of pyruvate kinase. The phosphorylation of pyruvate kinase leads to the inhibition of the enzyme. Furthermore, these reactions may be regulated by hormones via gene transcription. How? During fasting, as I said, low blood glucose level would be present. So glucagon would lead to decrease in gene transcription of the enzymes responsible for glycolysis to stop glycolysis and start gluconeogenesis. The opposite happens during well-fed state. Insulin increase gene transcription of the enzymes responsible for glycolysis, leading to increase of glycolysis and decrease of gluconeogenesis. Applications. We have already seen MODY 2 in the first lecture. We are going to go through it briefly. Known as maturity onset diabetes of the young type 2, characterized by impaired insulin secretion and hyperglycemia. It's a rare form of diabetes and it's caused by mutations of glucokinase, which was responsible for phosphorylation of glucose to glucose 6-phosphate. The second application here is arsenic poisoning. There are two types: trivinyl arsenic, which inhibits BDH complex, which is important for the link reaction, which we will discuss in the coming lectures. The second is the pentavalent arsenic. The arsenic competes with the phosphate group as a substrate for glyceraldehyde 3-phosphate dehydrogenase. We'll go back. This is the reaction which is inhibited. Once this reaction is inhibited, means there would be less glycolysis. Less glycolysis, so less ATP produced, so my cell would be deprived of energy, leading to toxicity. Water fluoridation. As we mentioned before, enolase is inhibited by fluoride. This would inhibit the anaerobic glycolysis of the mouth bacteria, which lead to inhibition of lactate production by mouth bacteria, decreasing the dental caries. Pyruvate kinase deficiency. This is a rare genetic defect of the enzyme. As you know, pyruvate kinase is very important for glycolysis. Any deficiency in this enzyme lead to decreased glycolysis, and as a result decreased synthesis of ATP. However, this would affect almost all the cells in our body, specifically RBCs. ATP is important for RBCs in order to maintain its flexible biconcave shape. So any decrease in that ATP, flexible biconcave shape is no longer maintained, and so the cell surface membrane is changed, leading to overall change in the cell shape, so that it is seen as non-self by the immune system cells, which are phagocytes and macrophages. Phagocytes and macrophages are going to phagocyte and lyse the red blood cells, leading to phagocytosis and lysis of the red blood cells, resulting in hemolytic anemia, because they have broken down the red blood cells. Energy yielding. As we mentioned before, we lost 2 ATP molecules and we gained 4 ATP molecules because of substrate phosphorylation, and we have produced 2 NADH. Go back to recap. First ATP lost, so -1. Second ATP lost, again -1, so -1 and -1 equals -2 ATP. And here 2 ATP by substrate phosphorylation. Don't forget that these steps are duplicated. And here again 2 molecules of ATP. These are 4, positive 4. And 2 NADH here. For substrate phosphorylation, 2 lost ATP, 4 gained ATP. So net ATP production by substrate phosphorylation would be 2 ATP, since 4 minus 2 is equal 2. Here we have produced 2 NADH. Each one of them will produce 3 ATP by the help of ETC. So 2 multiply 3, 6. 2 ATP by substrate phosphorylation, 6 ATP by ETC. Add them to get the total of 8 ATP molecules. Anaerobic glycolysis. During exercising, increased NADH to NADH ratio is seen, so NADH production exceeds the oxidative capacity of ATC, which means that the oxygen supply is insufficient. How the cells compensate this problem? By shifting from aerobic glycolysis to anaerobic glycolysis by conversion of pyruvate to lactate via LDH enzyme. Here lactate is the end product, unlike aerobic glycolysis where pyruvate was the end product. However, you are going to continue your exercise. You didn't stop. So, more anaerobic glycolysis is done, so more lactate is produced, leading to lactate accumulation. Lactate, once accumulate, as you know, lactate is acidic. This decreases the pH inside the cells, decreasing the intracellular pH, leading to cramps. You can no longer complete your exercise. You are fatigued. Lactate is dangerous. We need to get rid of it. How would we? Lactate is going to diffuse to bloodstream and reach the liver or the heart. In the heart, lactate is converted to pyruvate. Pyruvate would enter TCA and ETC and so on. In liver, lactate is converted into pyruvate, but here pyruvate has two pathways, either entering the TCA cycle or used for gluconeogenesis, energy yielding. Here we have 2 ATP lost, same as aerobic glycolysis, and 4 ATP gained, same as aerobic glycolysis. So net production of ATP by substrate phosphorylation is 2 ATP molecules. However, there is no NADH produced because I cannot produce NADH, because ATC cannot work, because there is insufficient oxygen supply. That's why anaerobic glycolysis lead to production of less molecules of ATP than aerobic glycolysis. Here is an application for the anaerobic glycolysis, which is lactic acidosis. What does lactic acidosis mean? High concentration of lactate in plasma. But when does this occur? In case of myocardial infarction, shock, pulmonary embolism, and uncontrolled hemorrhage. But why? In all these cases, oxygen supply decreases. Why? In myocardial infarction, your heart cannot pump the blood very well, decreasing the oxygen capacity reaching the cells. The same in case of pulmonary embolism. In uncontrolled hemorrhage, I'm losing the red blood cells, which are the transporters of my oxygen. So, not enough oxygen is reaching the cells. Again, low oxygen supply would lead that the cell would shift from aerobic glycolysis to anaerobic glycolysis, again increasing the lactate production, which finally leads to oxygen death. What does oxygen death mean? After stopping your exercise because you feel fatigued, you would realize that your breathing rate and heart rate would increase. Why? The increase to ensure that they increase the oxygen supply to the hepatocytes, because now hepatocytes are asked to convert all this lactate again to pyruvate. We need oxygen to help these hepatocytes. So heart and lung are going to pay oxygen debt for hepatocytes. And here is the end of our lecture. I hope you found it easy.